Good afternoon, dear colleagues that are following us mostly from Middle East and Asia Pacific area. For those who don't know me, my name is Nasser Al-Asmar, and I am Scientific Advisor and Embryology Director here at Igenomix headquarters in Valencia, Spain. Welcome again to an Igenomix webinar. In today's webinar, we will have an exceptional lecturer whom most of you will know. It is a pleasure for me to introduce my colleague and friend, Dr. Antonio Capalbo, Genomics Research Director and Laboratory Director at Igenomix Italy. And without further delay on my part, Dr. Capalbo, Antonio, please, whenever you want, the floor is yours.
Okay, thank you, Nasser, for the introduction, and welcome everybody to this webinar. I trust you can see my screen.
Yes, we can, Antonio.
Okay, that's great. Let me just move this, and I'm ready to go. So in the next 30-40 minutes, I'm going to walk you through this challenging topic, which is how we can detect mosaicism in embryos, how reliable it is, and what are the clinical predicting values of this type of designation that we can make during the course of an IVF treatment cycles with pre-implantation genetic testing for aneuploidies. So I always like to start my presentation by reminding the paramount importance about meiotically derived aneuploidies for human reproduction and fertility. We all know very well from decades of investigation in the genetics of pregnancies or in postnatal or prenatal space that aneuploidies are mostly coming and occurring in humans as a consequence of a defective meiotic segregation, which is extremely linked to the female age. And recently, a large group of investigators that was led by Professor Eva Altman reported this data on thousands of human eggs that were analyzed across the board of reproductive lifespan on the female from the menarche to the menopause. And they have actually characterized the aneuploidy rate in thousands of oocytes that are, and the euploidy rate is displayed here in this U curve, red curve here. And you can see how this curve is actually shaping the red line on the bottom, which is showing the human fertility over reproductive lifespan, meaning to say that the errors, the chromosomal errors in human eggs are really the ones that matters for reproduction, that are shaping human fertility over reproductive lifespan. So when we have meiotic aneuploidies that are present in a zygote and in a developing embryo, all the cells are supposed to be affected by the same chromosomal abnormality. And this is not posing any issues on a diagnostic perspective, because wherever we take the biopsy, then we're going to get a good representation of the remaining chromosomal constitution of the embryo. And we also know very well that when an embryo is aneuploid because of meiotically derived aneuploidy, then the outcome is very well described, and we can provide a very good counseling to patients about the fate of these embryos, that is implantation failure, miscarriage, or in very few situations it can be an aneuploidy which is viable and compatible with life, and this will lead to an aneuploid conception. Now, we often hear in some meeting that this type of aneuploidies can get through self-correction during pre-implantation development. However, this is more a myth rather than a fact. We all know very well that self-correction of a meiotically derived aneuploidy is an extremely rare event, and we know that because this event is measurable. We all know well that when a trisomy that is inherited meiotically is corrected throughout embryonic development, this will lead to a population of cells that have a uniparental disomy. And uniparental disomy at the blastocyst stage has been measured in the past on thousands and thousands of human blastocyst stage embryos by the group of Nathan Treff and colleagues, showing that this event is extremely rare, 0.06% of human blastocysts have been shown by SNP array to have uniparental disomy, meaning to say that self-correction of meiotically derived aneuploidies is very rare, and once an embryo has a meiotically derived aneuploidies, this is very associated with a very clear negative outcome in case these embryos are transferred. And we know that also from many different clinical studies, particularly from non-selection studies that are, I think, the most powerful design in order to estimate and evaluate what is the positive and negative predictive value of a PGTA assay and analysis that you can perform. Particularly in this design, you can see that what usually happens is that you take a biopsy, you save this biopsy for later stages, aneuploidy testing, and you transfer the embryos without knowing the aneuploidy status. So after the transfer, the trophectoderm biopsy is analyzed usually in a blinded way, and you can really evaluate with very few or minimal level of errors what is the positive predicting value or the negative predicting value. The positive predicting value is how many aneuploid embryos are indeed aneuploid and they are not producing a live birth. The negative predicting value in this case is how many euploid embryos will result in a live birth. These are very valuable and important information to evaluate what is the accuracy and the clinical validity of our assays. And as you can see here in this study, out of the 102 embryos diagnosed as uniform aneuploidy, none of them progressed to a sustained implantation or delivery, while the negative predictive value was very high, with 64% of euploid embryos resulting in a live birth. We know that this topic is very controversial, and indeed, time to time, we also read throughout the literature some discordant findings. For instance, if you have the chance to read this paper by the group of Norbert Glaser, you could see that at the beginning they were complaining and they were questioning the accuracy of aneuploidy testing because they show that some transfer of fully aneuploid embryos resulted in live births. However, looking carefully at the data that they have produced in the original version of the National Cell Biology paper, we could make argue about how these data were delivered, because at the end they were inconsistent with previous publication and presentation of this data set, and they make an amendment recently showing that indeed out of the 61 fully aneuploid embryos that were transferred, none of them they result in a live birth or in an ongoing pregnancy. Particularly, they also observed that many of them result in miscarriage that had the same abnormality that was previously detected at the pre-implantation stage. Now, today we have many of these studies where the outcomes of uniformly aneuploid embryos have been evaluated, usually blinded and with a prospective non-selection design. We have recently collected all this evidence and put it in the same table, in a single table. And as you can see here, the lethality rate of embryos that are designated as fully aneuploidy following a PGTA, the blastocyst stage, with many different technologies and assays for aneuploidy testing is about 98%, meaning to say that the vast majority of these embryos are not going to make a baby. Most concerning outcome following the transfer of fully aneuploid embryos is, however, the miscarriage rate. As you can see here, based on the evidence available today, if you decide to transfer an embryo which is uniform aneuploid, you should expect to have 86% of miscarriage rate. So I think that today we have a robust and reproducible evidence about the fact that uniformly aneuploid embryos are very well designated as normal and shouldn't be considered for clinical use. Now, on the top of euploid and aneuploid embryos, we also have an intermediate category, an hybrid category, which is the so-called mosaic abnormality. In this case, the aneuploidy originate during the mitotic division of the embryo and will generate a developing embryos which is made up of a population of normal and abnormal cells, which is of course causing some issues and posing some challenges for that diagnostic perspective, because by definition it will be very difficult to make an accurate evaluation of the mosaicism rate by analyzing only a portion of the whole embryo. Now, to make a little bit of clarity around the terminology and also to buckle some myths and misconceptions on this topic, let's start first from the definition. So mosaicism refers to the presence of genetically distinct cells within an organism, in our case an embryo, that result from post-zygotic maturation event. We may have two different types of mosaicism. One is the genetic mosaicism, where we have that the variation happen at the DNA sequence level, such as single nucleotide variants or indels, or we may have cytogenetic mosaicism, where the variation and the abnormality is at the chromosomal level. So, and then further on that, the cytogenetic mosaicism is the one that we usually discuss, and we have been discussing over the last years in PGT, can be further distinguished and break down in the aneuploid mosaicism, where we have distinct aneuploid complements due to a mitotic error that happen in an embryo that is already affected with a meiotic aneuploidy. So at the end, this is not a big issue because all the cells of this embryo will be abnormal. But the one type of mosaicism which is concerning us the most is the diploid aneuploid mosaicism, where we have a combination of normal and abnormal cells. So further on that, we have also some misconception around mosaicism when we hear that since we are all mosaic in adult stage, all human embryos, human preimplantation embryos should at the same level be mosaic, but this is a very weak assumption. I mean, it's mostly speculation, because we all know well that in our adult life we accumulate mutations. So it makes sense that we are all mosaic at adult stage, but it doesn't mean that also all the embryos should be mosaic at the chromosomal level. It may be the opposite can be true, because I mean mosaicism is linearly correlated with a number of mitotic division that happen within the body, and I mean, to be fair, once we reach the blastid stage, few hundreds of mitosis happened at this stage, and that are significantly lower as compared to the postnatal stage, for instance, or the post-implantation stage, where millions or billions of mitosis happen in our cells. So if we wanted to see that mosaicism is really linearly correlated with the number of mitoses that happen from the conception time point, then probably the pre-implantation stage is at least subject to mosaicism as compared to later stages of our life cycle. Another important point to make clear before we go through mosaicism in PGTA is the incidence of mosaicism in human pregnancies, which is being estimated in hundreds of publications and works in the past. And we all know well that mosaicism is affecting about 0.3%, 0.4% of all pregnancies. And in particular, we also know very well that IVF is not a risk factor for mosaicism. There have been several studies that have compared incidence of mosaicism in IVF and spontaneous pregnancies, showing that they happen at similar rates, meaning to say that infertility by itself or the whole IVF procedure that we perform in our IVF treatments are not increasing the risk for mosaicism in pregnancies. And as a consequence, we are not under a big pressure of increasing our diagnostic capability towards mosaicism detection in PGTA treatment cycles. Now, let's have a look at how mosaicism is detected in PGTA cycles when we analyze trophectoral biopsy samples. Usually, what we commonly do based on standard NGS sequencing analysis protocols for PGTA is to report mosaics when we see intermediate chromosome copy number, that are those values of copy numbers following between the normal and the trisomic or monosomic range. So when we see this pattern that is displayed in the slide, we usually refer it as intermediate copy number, and this is what is used as evidence to report that that biopsy is mosaic for a given chromosome. Now, this is extremely different from what is done, for instance, in prenatal diagnosis when we have when we do CBS or amniocentesis, and we can visualize different cell population by looking at metaphase spreads, and when we can really see that there is a population of cells that are normal in a sample that is also made up of normal cells. In this case, we use these algorithms and intermediate copy number to call mosaicism, and this is subject to many different assumptions and weaknesses. For instance, we know that very well that intermediate copy number can also arise as a consequence of technical variability or experimental noise, and these may come as a consequence of the fact that working on a clinical setting, we may have biopsies that have a different quality, different cellularities, and this is of course something that may introduce some technical variation and generate some artifactual intermediate copy number. Another weakness of this approach is that usually the thresholds that are used for calling mosaicism are those that are developed on cell lines mixer models, where mosaicism is mimicked by mixing up normal and abnormal cells in different proportions, and by deriving the thresholds and sensitivity and specificity versus some specific thresholds for mosaicism classification. Of course, I don't have to tell you that working on cell lines is completely different as working the standard clinical activity that we perform on clinical biopsies, because cell lines are, of course, a more reproducible and standardized study model as compared to what happened and to the experimental variability that we are exposed to in a clinical setting. Now, this consideration, this technical consideration, clearly explain the reason why some clinics over the last years have been reporting some very low rates of human embryos being mosaic at the blastocyst stage, 2 to 3, 4%, while other clinics have been reporting 40%, even up to 30, 40% of human embryos being mosaic. Of course, I don't have to explain to you that it's not the biology which is changing from one clinic to another, but it's more a matter of how you can control the experimental variability and how you are capable of lowering the overcalling of this type of abnormalities coming from experimental variability, and this is almost all there. So a second assumption that is very weak for reporting mosaicism in pre-implantation human embryos is the sampling bias. As you can see here, I think it's very clear that by definition, if an embryo is a true mosaic embryo, then depending on where we take the biopsy, we may get completely different outcomes. And this is something that, of course, always need to be acknowledged when performing any type of diagnosis, that we are, limiting our observation to a sample that may also not be representing the remaining chromosomal constitution of the embryo in both ways. It may be that we take only euplant cells while the embryo is mosaic, is a true mosaic, or it can happen that we take all the aneuploid cells while the embryo is a true mosaic. But even if we sample the cells, a trophectory biopsy, which is a mixture of normal and abnormal cells, the rate of mosaicism that we can identify in this biopsy can be completely different to what then is in the embryo. Now, an important point is that trying to lower the overcalling of mosaicism, because when we report mosaicism in the clinical setting, this comes with very relevant consequences for the couples undergoing the IVF. First of all, we need to acknowledge that when we report a diagnosis of mosaicism, the couple will have to undergo an additional session of genetic counseling, and they will have to evaluate the embryo transfer of these mosaic embryos in a context of increased anxiety, distress, and of course, this is not something that should be, I mean, generated by uncertainty, and by the uncertainty of some types of diagnosis. Then the second consequences that the couple will face when transferring an aneuploid embryo, a mosaic embryos, is the increase or the indication that has been generated to go through an amniocentesis procedure, an invasive procedure that comes with some very well-known risk of iatrogenic abortion, and that should be, of course, justified by some evidence, some clinical evidence about the positive predicting value of this type of mosaic diagnosis. But I think that the worst outcome of mosaicis reporting is the fact that most of the patients in the past have been discarding these embryos. If we look, for instance, at the publication of Moon and colleagues in 2017, you can see that less than 3% of the almost 5,000 embryos that were designated as mosaic, then they were transferred back to patients, which is highlighting a very concerning practice and a very alarming risk of wastage of embryos that are otherwise healthy and capable of a normal implantation and development to term. So what is this practice based on? This practice of designating embryo as mosaics in the broad spectrum of 20 to 80% of variation was supported only on retrospective evidence about a slightly lower live birth rate outcomes following the transfer of these low mosaic embryos or high mosaic embryos. There is the largest studies published by Manuel Viotti and some other colleagues that look at these outcomes retrospectively. They found out that the low mosaic category had just a slightly lower live birth rate potential, while the high-grade mosaics, they were indeed associated with a significantly lower live birth rate. Now, there is a very important bias when looking at this data retrospectively, and the big bias is the one of population selection, because in the standard clinical workflow, when reporting mosaics embryos that are designated as mosaic, they are left behind. They are transferred usually as last option after the transfer of the euploid embryo that goes first. So the situation here is pretty much clear that when you downgrade and they prioritize embryos that are mosaic, you get all the good prognosis patients getting pregnant with euploid embryo transfer, with the first, the second, or the third euploid embryo transfer, while the patients that failed with previous euploid embryo transfer are the ones that are receiving the mosaic embryos, and the ones where we are going to measure what is the clinical gain of a mosaic embryo transfer. So in this case, we are it is very clear that when we look at this data respectively, we are measuring the reproductive potential of mosaic embryos on a very poor prognosis population of patients that have been selected because they failed with one or multiple previous euploid embryo transfer. So what we did in our study was to trying to avoid this kind of selection bias, and to do so, we have designed a prospective non-selection trial back in 2017, where all the putative mosaic embryos between 20 to 50% of intermediate copy number were reported back as euploid. This has helped to avoid the selection bias because the embryos for transfer were chosen irrespectively to the genetic outcome. So both the physician and the patients were blinded to the outcome of the mosaicism in the 20 to 50% of variation, because these embryos were indeed reported as euploid in the same way as for the fully euploid embryo that did not show any intermediate copy number. So after this, during the course of this trial, after the transfer, we were able to unmask and unblind the results of the genetic testing, and we could generate 3 different study group and arms here: the uniform euploid embryos, the low mosaic between the 20 to 30% of variation, or the moderate mosaic category between the 30 to 50% of variation. And we could analyze the clinical outcomes as well as do some postnatal follow-up on some of these cases that resulted in a live birth. So just to make 100% sure and clear to you, embryos in the low-grade mosaic or in the medium-grade mosaic were all reported back to patients as euploid in order to avoid any population selection bias. The first step in our study and how we set out the thresholds for reporting these embryos as normal, this was based on a rebiopsy analysis of 73 different blastocysts that were donated for research and were disaggregated down to 5 different portions, 4 trophytoderm samples and the ICM. So in this biopsy study, multifocal analysis, we could observe, for instance, what was the positive predicting value of a low mosaic versus the ICM or the remaining embryos. Here we have an example where a low mosaic in a clinical trophytoderm biopsy was not confirmed in subsequent biopsies. Or you have here another example where a uniform aneuploidy for chromosome 21 was indeed confirmed across all the biopsy of the same embryo. And this way, we were able to evaluate what is the positive predicting value and what were also the risks of having these low or medium mosaic embryos being classified as euploid. Now, this is kind of complex data to show in a slide, but I will try to walk you through this table, which at the end you can read it very well. So how this table works? This table works with a reference trophytoderm outcome on the left side, and with a confirmation rate on the subsequent biopsies that were taken in that embryo. In the case where you have a normal ICM on the left side and a normal ICM on the right side, and also according to the other trophytoderm outcomes of other trophytoderm biopsies. So just to give you a very simple example, when the reference trophytoderm outcome, the reference trophytoderm biopsy was fully aneuploid, we could see that 98% of the embryos, and indeed all biopsies, all the remaining biopsies, fully aneuploid as the original ones, including the inner cell mass, while in 2% of cases, that biopsies was present only on the reference trophytoderm outcome. So it was most likely a very confined mosaicism case. If we go to the high mosaic category, which is 50 to 70% of intermediate copy number, you can see that this is really the gray zone of our diagnostic pipeline and assay, because here you have that 65% of the high mosaic classification turns out to be fully aneuploid, while the remaining 35% are indeed some sort of true mosaic embryos and true mosaic pattern. However, it's important to know that the high mosaic category, at least in our diagnostic pipeline, they account for 1.6% of all embryos and diagnoses that we make at the blastocyst stage. So even if we have some level of uncertainty here in this category, is not going to impact the clinical outcomes in a significant way. And most importantly, usually we discourage and we do not recommend the transfer of these embryos, as in vast majority of cases, they are indeed fully aneuploid. So if we look then back to the euploid, low mosaic and medium mosaic, you can now appreciate that all these patterns, they show a similar confirmation rate throughout the embryo, meaning to say they had the similar risk of being aneuploid. And therefore, we consider that these embryos could be treated as a single category of euploidy. Now, here you have the table with the demographic and cycle data of the enrolled patients. We could enroll 783 patients, 824 stimulation cycles. The mean female age is consistent with advanced maternal age population, as well as all other demographic and cycle data. The unemployed rate was the unemployed rate was 41%, including the fully unemployed and high mosaic embryos. In this study, 5 different centers in Italy were involved and performed all the recruitment and clinical procedures, namely in particular Genera, Humanitas, and Demetra, that of course we want to really acknowledge and thank for their contribution in the study. Here you have the clinical outcomes, the ones that really matters, and you can immediately see that the outcomes of the 3 study groups were similar in terms of positive pregnancy test, biochemical pregnancy loss, miscarriage rate, and live birth rate. You really cannot see any different. We couldn't see any different. We reached our planned sample size. We performed a multivariate logistic regression analysis in order to control for confounding factors. Even by a multivariate analysis, we did not see any association between the PGTA category, whether it was euploid, low mosaic, or medium mosaic, to the clinical outcomes. Also, neonatal outcomes and obstetrical outcomes were similar between the 3 groups. We've been trying to develop other definitions of mosaicism, to take new parameters, to try to link any kind of form of mosaicism to clinical outcomes, like the mean mosaicism rate, the mean number of chromosomes that were in mosaic, the incidence of the so-called complex mosaic, more than 3 or more than 5 chromosomes being involved in an intermediate copy number, but we fail to detect any statistically significant association with the principal clinical outcomes that we were investigating. Here you have a representation, a figure showing all the mosaic chromosomes that were transferred during the course of our trial, whether they were classified as low or medium mosaic. And as you can see, since the study was very well controlled and blinded, we had also the transfer of embryos with mosaic trisomies for chromosome 21, 18, 13, or other more risky chromosomes. But we never got any issues related to that so far. So as a last analysis in our study, we also perform some level of prenatal follow-up. In particular, in our clinical practice, we do not recommend invasive PGT for all cases and all women conceiving by euploid embryo transfer, because we still don't have any evidence that the risk of aneuploidy is higher than the invasiveness of the procedure. So we could observe and follow up only those cases that decided to go through amniocentesis after the transfer of euploid or putative mosaic embryos in our trial. And as you can see here, the ones that we had a follow-up, cytogenetic follow-up during the prenatal diagnosis, the only case of confined placental mosaicism was detected following the transfer of fully euploid embryo. That was a confined placental mosaicism with 20% of abnormal cells with a trisomy for the chromosome 22 that was then shown to be normal at the amniocentesis. However, we could collect some saliva samples from newborns that were obtained following the mosaic embryo transfer. In particular, in this phase of our study, we could collect saliva samples and DNA from the parents in order to investigate if these babies that were born following the mosaic embryo transfer had any evidence or instance of mosaicism or UPD, at least as investigated through saliva samples. And to do that, we run a high-density SNP array. We use some bioinformatic, well-established and validated bioinformatic algorithm to check for mosaicism and UPD in these newborns, but we fail in this small data set to detect any evidence of mosaics or UPD following the transfer of mosaic embryos or putative mosaic embryos. And finally, what I wanted to show you is this modeling that we did in the course of our trial, assuming the situation where low mosaic or low and moderate mosaic embryos would not have been transferred. So the case where we exclude all the pregnancies and the live births coming from the transfer of mosaic embryos. As a result, if we make this modeling, we could observe that over 1/3 of babies would not have been born if these mosaic embryos were not transferred in this trial, which make the case for a significant loss in terms of cumulative live birth rate that you can get if you do not transfer or consider for transfer these embryos. So these evidence are confirmed and corroborated by independent studies and findings. For instance, in this study, Mina Popovic and colleagues, they find out that when growing embryos up to day 12 and comparing the outcomes of a trophectoral biopsy and this embryonic outgrowth on day 12, they could get 100% concordance rate for both the uniform euploid and aneuploid embryos, while the positive predictive value of a mosaics consistent finding in trophectoral biopsies was extremely low. And also, we have seen in animal models and modeling of mosaic embryo transfer, this is not actually representing what happens in mosaicism, because these were chimera embryos. But in this case, for instance, Helen Bolton and colleagues, they observed that abnormal cells following the transfer of chimeric embryos, they get lost in the post-implantation stage, and they can make normal babies, as well as in a more recent Nature paper by Purans and colleagues, they show the existence of early developmental bottlenecks in human placenta that are able to normalize post-zygotic mosaic aneuploidies in human embryos. We actually don't know whether all the live births that we obtained, they were a kind of false positive diagnosis of mosaics because of technical variability or experimental noise, or whether they were true mosaics that is extremely confined to a small region of the placenta or the trophectoderm, and then they get lost in the early post-implantation stages. But both explanations are valid and are worth to be further explored in future studies. What I want also to show you is that what are the risks of transferring mosaic embryos or apparently mosaic embryos following PGTA. They are extremely low because you can see here in this table that Nathan Treff has developed and pulled out data from many different studies. You can see that today we have only one case where the same mosaicism was detected in the PGTA stage and confirmed later prenatal diagnosis stage. This baby didn't show also any phenotype associated with the mosaicism, and therefore today we for sure do not observe any kind of increased risk in mosaicism following the transfer of embryos that are designated as mosaic at the embryo, the pre-implantation embryo level. For sure, there will come new cases where we observe mosaicism following the transfer of mosaic embryos because this practice is increasing. PGTA, the adoption of PGTA is increasing, so we will have the follow-up on these embryos is higher and is more detailed as compared to the transfer of uniformly euploid embryos. But important point for the future will be to compare what will be in a very systematic and standardized way, what is really if there is any additional risk of having a mosaic pregnancies following the transfer of an embryo that was designated as mosaic, and because it showed an intermediate copy number value. So at present, I think we have vast majority of the evidence suggests that there is not increased risk as compared to the transfer of fully euploid embryos, because indeed what we have also seen in other studies, for instance, in this study by Frendal, Monet, again, Grifo and colleagues, is that you can also find many cases of mosaics following the transfer of fully euploid embryos. In this study, although based on a very small sample size, they had 57% of product of conception showing mosaic abnormality following the transfer of fully euploid, uniformly aneuploid embryos, also using high resolution next generation sequencing. So the point is that always important to cancer patients about the fact that when you, even if you're transferring a uniformly euploid embryos, you should also expect some minimal level risk of having mosaic pregnancies as for the general population. The very last point, my presentation is about also this practice that has been generated with the introduction of mosaics reporting in PGTA cycle, that is the prioritization of euploid embryos over the mosaic, regardless of any other embryological parameter, that is, morphology, day of development, and other morphological features that have been very well established and are associated with clinical outcomes from decades. So there were at the beginning a couple of scientific societies, the PGDIS and the COGENT, were recommended to transfer euploid embryos first, regardless of morphology. And I think, and I'm pretty much sure that there is no study or no evidence today to support this recommendation, in the way that I'm pretty much sure that there is no data telling us that transferring a bad quality euploid embryos is a better option for patients than transferring 20% mosaic embryos of good quality. So this is something that has been indeed corrected in more recent guidelines, and it is something for sure where we need to take into consideration different factors, but in particular morphology that we know that has been associated for a long time in embryology to clinical outcomes. So in conclusion, intermediate copy numbers, the ones at least below 50% in clinical trophectoderm biopsies, are either technical variations of the methodologies and technologies we're working on single cell or equivalent type of samples, or there may be true mosaicing that is confined to a small trophectoderm portion without ICM involvement and implication for the pregnancy. As a consequence, we believe that reporting mosaics in this low to moderate range has no elements today of clinical utility, while can result in a significant wastage of viable and otherwise healthy embryos in case the patients perceive these embryos as chromosomally abnormal and they will discard them for clinical use. I think it is extremely important in this study to properly validate by non-selection trials and randomized trial the PGTA assays, because this can really give us the true predicting value, clinical validity and utility that these technologies can bring us and can bring to our patients. So, and finally, I want to acknowledge all the colleagues that have contributed to this non-selection trial. It was a big effort. It took 4 years, but at least we are very much confident that this will help the clinical practice of PGT today and in the near future for the benefit of patients. So that's all on my side. I thank you all very much for following the webinar, and of course I'm available for any questions that you may have. So please do not hesitate to address questions.
Thank you very much, Antonio. Brilliant as always. And as Antonio said, I would like to invite all of you to make your questions now. Don't be shy, I won't say the names, by the way. But Antonio, I feel like every time that I can see you lecturing, even though same topic, 3-4 weeks of differences, I feel with more power, with more knowledge. So thank you very much. Thank you very much about that. And well, there are some people asking if they can ask the questions in the question box, and the answer is yes. Please go ahead, Let's start with the first one, Antonio. We have around 10 minutes in order to make this Q&A session. We were afraid to transfer mosaic embryos. So question is, should we be afraid right now after the data that you showed us? I guess it's about proper genetic counseling.
Well, I think that first of all, you should get to know the validation data that your genetic laboratory. You should ask your genetic provider to show what are the validation data about the use of intermediate copy number to designate mosaics, what type of experiment they have done, and what type of validation data they are available in order for you to make the best consideration for your patients. I understand and I appreciate that not all providers have developed non-selection studies, so you may have work around that by looking at biopsies data or even cell lines, even though this is not the ideal design and setting. But still, you should be aware about that, and you should consider that most there is quite a large experience, and most of the mosaics below the 50% can be, I think, from most providers, can be safely considered as normal and as for transport, yes, and can be considered for embryo transfer without that many concerns.
Thank you, Antonio. There's another one that says, in your opinion, should we treat mosaicism different depending on the chromosome? How about differences between low and high mosaics?
Yeah, low mosaic, most of these low mosaic embryos, they can be completely normal embryos just by technical variations. So it's really, I mean, you can safely consider them as normal, of course, and as I said before, I mean, ask data about validation. High mosaic, we're getting to know, and we're seeing that most of these high mosaic copy number values, they can come as a technical variation from the fully aneuploidy spectrum. So I won't recommend to transfer these embryos. And because, I mean, there is very high risk that you are then transferring fully aneuploid embryos. So everything should be, I think, should be properly validated. We should take this decision based on validation data, clinical and experimental data. For instance, now we know that up to 50% of variation, there's no big issue in transferring these embryos because we have evidence from a non-selection trial and from hundreds and hundreds of embryos that have been disaggregated and investigated. And I think that in order to make this decision, you should have similar evidence from your provider in order to make the best decision for your patients. So now we know that we can safely transfer the low to medium mosaic embryos, and that the vast majority of the high mosaic signals are indeed coming from uniform aneuploid embryos. And we can make best decision for improving clinical utility of the patients. The point that I expect in the future is that that once we want to develop new algorithms, we want to introduce new diagnostic criteria in PGTA. We first perform this kind of studies before introducing big changes that are not validated and are not supported by evidence into the clinical practice. And years later, we realized that probably this was not the best way forward. So I'm just hoping that for the future, new developments will be better validated before being used and applied to patients.
Thank you very much, Antonio, because there is a lot of questions regarding about the high mosaics and also specific chromosomes, you know, such as 13, 18, and 21. What should we do? If we should transfer right now, we should wait until further studies. non-selection studies, because well, we will talk later also that we're asking about this bias of another studies that because you transfer first euploids. But yeah, regarding, let me, well, you told us at the very beginning of the lectures, but also there is a lot of people asking about, again, self-correction in embryos. So I guess that it is worthy that if you can elaborate a little bit, like you explained us at the very beginning of the talk, what do you think about this self-correction? Apparently, that a myth, right?
Yeah, sure. I mean, self-correction is something that is very well known in genetics from decades, and is something that indeed is may happen, and in particular, but we need to be clear, because this terminology is often used in a wrong way by many critics of PGTA, and is often, I mean, used and reported as an evidence against PGTA, which is not. Self-correction is by definition when an embryo that starts as fully aneuploid, like a trisomy, because the oocyte was aneuploid, that's been the case, then goes through self-correction during the pre-implantation development, and becomes diploid at the blastocyst stage. But the self-correction event by itself, so the generation of a diploid line, may bring the fact that some of these self-correction events will result in a uniparental disomies because of the two chromosomes that were inherited from the oocyte. They may stay into the corrected cell lines, and we can see it. We can analyze uniparental disomy in blastocyst stage human embryos, and there is a very large study at that time from the RMA group that was led by Nathan Treff, and they show that indeed UPD at the blastocyst stage is very, very rare. That is telling us that these self-correction events very rarely happen in pre-implantation embryos. So what we know, and the mounting evidence is showing, is that when you have embryonic aneuploidy in the embryo, this is fixed throughout development, and is of course associated with negative outcomes for the patients in case these embryos are transferred. So the self-correction event is measurable, to answer to your question, and has been measured in thousands of human blastocysts, and has been shown to be extremely rare. I hope now this is clear.
Thank you very much, Antonio. There's a question that I guess that it's more to a specific lab, but I will make to you just to check your opinion. They said the question was, can we select non-mosaic workflow for PGTA analysis? I mean, yeah. I guess the question is more than, you know, that, okay, I don't want to know about anything mosaic, let's make it aneuploid, but I would like to ask you about what would you recommend?
Man, that's a very good question. I mean, for instance, my experience, I never reported a mosaic embryo, so the answer is quite easy. I've been always using this kind of binary way of reporting, which is euploid and aneuploid. And to be honest, I never had the experience so far of mosaic pregnancies, as we probably never had in the course of standard IVF. So it's possible to report embryos as euploid and aneuploid, and this is bringing also many advantages, I think, for the clinics and for patients that do not have to face with uncertain results, with uncertainties, with additional costs for genetic counseling, wasting time, increasing anxiety of patients. I mean, I have had many patients asking for advice and calling me when they got pregnant following the transfer of what was designated as mosaic embryos, and they didn't, I mean, they were not enjoying their pregnancies at all. So I think before introducing all this level of uncertainty, and before I mean, delivering diagnosis of chromosomal abnormalities like mosaicism, we really need to have stronger evidence. And yes, it's possible, of course, to have a binary approach, reporting only euploid and aneuploid embryos, drawing the threshold on in the middle. Then there are, of course, chromosome-specific considerations. We all know well that from a technical standpoint, some chromosomes have different amplification profile than others, but in principle, it's possible to apply the same concept to all chromosomes and drawing the line in the middle and reporting only the euploid and the aneuploid embryos. And I think that this is also the most accurate way to report in PGTA today, based also on rebiopsy studies that are indeed showing that this is probably the most accurate way of reporting.
Thank you very much, Antonio. There is another question regarding trophectoderm biopsy that are asking you which is the best site for taking trophectoderm biopsy. I don't know if you're trying to avoid mosaicism with the biopsy.
No, I mean, I think that reducing or lowering the level of mosaicism can be achieved through the improvement of the biopsy, but not because it's biological phenomenon, but I think it's really important to standardize. This is one of the most critical things in the whole workflow, standardize the biopsy in order to get consistent samples that are of similar cellularity, of similar quality, that have been processed in a similar way. This is really what matters in order to get clear molecular profiles and also to be I mean, able also to get good quality data and good diagnosis from the genetic laboratory, and also to lower all this level of mosaicism or overcoding of mosaicism, at least the ones that is caused by experimental variation. So focusing on the IVF lab, on the biopsy and methodologies that are used and that can be properly applied and standardized, this is, I think, really crucial in PGTA.
Thank you again, Antonio. So we are almost at the end, and let me ask you about this question that I was mentioning, that you were talking about patient selection bias. So we transfer first the euploid ones, and then at the very end, the mosaic. So obviously the patients were of poor prognosis at the end, right? So, and also you showed us one of the last slides regarding this, right? Eventually, do you think that will be important morphology? I mean, maybe mosaic embryo grade A could be better than a euploid embryo grade C or D. So what do you think? This is some of the questions that we are receiving right now.
Yeah, indeed. This is also a good point, Nasser. I mean, there are no studies comparing the two approaches. While there were recommendations saying and telling us to do that, so to transfer even better quality euploid embryos over the good quality apparently mosaic embryos, which for me was kind of shocking to see how recommendations were delivered without any evidence in support. So at present, I think if you are receiving mosaicism reporting and the mosaicism is in the low category, I think morphology should be considered together, or should be even the main consideration, because at the end, for the value of mosaicism, the clinical value of mosaicism, we have evidence, we have very robust clinical evidence from decades of application in IVF, while for this mosaic classification, there is very little evidence that is improving any of the outcomes of patients, unless we are not talking about high mosaic, which is more similar to a uniform aneuploid.
Yeah, totally agree. That I think that in these days we will take into account all the different approaches, and maybe morphology would be one that it's going to be more interesting for us in order to make the decision to perform the transfer. So Antonio, prior to say you goodbye, I would like you to have your 1 minute golden minute, I would say, in order to have a take-home message for all the audience that are in here waiting for your last opinion.
Well, thanks, Nasser. So I think the final message is, I mean, to be reassured about the clinical use of embryos that are classified as low or moderate mosaic. because this can be either a technical artifact, or it can be the presence of some abnormal cells that are extremely confined to a small portion of the developing embryo that are not affecting the inner cell mass and not affecting also the pregnancy. So, I mean, of course, we need to keep focusing our attention to expand our research and investigation in this field, get more data, but I think the message, the final message, is that we can be reassured and we can consider more confidently the transfer of these embryos and the clinical use of these embryos that otherwise may significantly impact the chance of patients to get pregnant in the course of an IVF treatment cycles, which is really what we do not want, lowering the cumulative live birth rate because of PGTA or overcalling that are made in PGTA.
So thank you very much again, Antonio, for the wonderful lecture. I will send to your way the questions that we didn't make it, we didn't have to make it. And thank you all for being here today again with us. It's been a pleasure to have you for all of our webinars, and we will wait for you on the next one. Thank you very much and have a great day. Thank you, Antonio, and thank you all.
Thank you so much. Thank you.